Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, in previous lecture, we have already initiated with the topic that is non-allelic gene interaction, and in that two gene interaction we have completed, in which we have studied already complementary gene, duplicate gene, epistasis, supplementary gene, polymeric gene, additive gene, and collaborative gene interaction. In continuation of that, today we are going to understand multiple gene inheritance. So the topic is polygenic inheritance. Now polygenic means multiple gene. So we are going to discuss here multiple gene inheritance, how multiple gene get inherited. Okay. So if we discuss in more detail, then in this polygenic inheritance, in this polygenic inheritance, more than one non-allelic gene, more than one non-allelic gene regulate the expression of character. Regulate the expression of character. Means multiple gene means gene in a quantity they are affecting a multiple gene, or a gene in a quantity they are affecting the particular character. So it is also known as quantitative inheritance. Okay. Now quantitative character means what? In this character, we will see that each dominant allele each dominant allele each dominant allele have a fixed proportion of expression for character. Means dominant allele either of any gene, if it is in a dominant form, it has a contribution for the expression of gene, so it is in contributory in nature. Whereas if we talk about a recessive allele, then recessive allele have no contribution in expression of this trait, so they are non-contributory in nature. So whatever the gene, if we are taking the three genes, that is A, B, and C, if any is dominant, A is dominant, B is dominant, and C is dominant, they have an equal expression, equal proportion of expression. And the total, whatever the total number of gene, whatever the allele, whatever the gene, the total number of dominant allele will contribute for the expression of gene or expression of a character. So means the expression of a character the expression of character depends on number of dominant allele. Depends on number of dominant allele, and their expression is very much affected by the environment. So let's understand actually what is this. For that we have to take an example and then only we can discuss. But for before discussing the example, here we are going to talk about some of the initial history. Then it is known that father of polygenic inheritance, if we talk, then they are then he is J. Collerenter, and he worked on tobacco. But detailed study of polygenic inheritance is done by or carried out by Nilsson and LA on wheat and tobacco plant, on kernel color of wheat and corolla length of tobacco plant. Okay, so by taking this two example, he studied in detail this polygenic inheritance. In kernel color of wheat, if we take, then kernel color in wheat, it is controlled by two genes. And when it is controlled by a two gene, if we see its color, then it is dark red, and the intermediates are obtained, and in a recessive the white is obtained. So when all are dominant controlled by two gene, suppose that is gene A and B, when all are dominant like capital A, capital A, capital B, capital B, then the maximum expression of all gene will occur, and that will produce a red color, and that is a dark red. But when small b is there, they are not contributory in nature, so minimum expression will occur, and the white kernel color is obtained. So if you make a cross of this, other variation can be observed. So let's make a cross. We are taking a parent generation, and in parent generation we are taking a parent that is dark red and crossing it with the white kernel color. Dark red genotype will be capital A, capital A, capital B, capital B, and white will be small a, small a, small b, small b. The gametes which are going to be produced in this, that is are capital A, capital B, small a, small b, and when this gamete fuse, they are going to produce capital A, small a, capital B, small b, that is F1, and it is intermediate red. Now when we make a selfing of this, then we again cross this intermediate red, capital A, small a, capital B, small b, with the capital A, small a, capital B, small b for the F2 generation. So how the cross of these two will occur and the next generation will be obtained, that we will discuss in the next slide. So here, when we take a capital A, small a, capital B, small b, the gamete are going to be capital A, capital B, capital A, small b, small a, capital B, and small a, small b. Suppose these are the gametes of a one particular plant, and we are taking it as a male plant. Now the gametes of female plant we will write here. Here also female plant will have with the genotype capital A, small a, capital B, small b is going to produce the this same four types of gametes. Then it will produce a offspring with the capital A, capital A, capital B, capital B, capital A, capital A, capital B, small b, capital A, small a, capital B, capital B, capital A, small a, capital B, small b. Here, by fusion of these two, the capital A, capital B, small b will be produced. Capital A, small b is there. Capital A, small a, capital B, small b is there. And capital A, small b is there. Then capital A, small a, capital B, capital B, capital A, small a, capital B, small b. Here also you will find capital A, small a, capital B, small b. Here capital A, small b, small b. Here small a, capital B, capital B. Small a, capital B, small b. Small a, capital B, small b, and small b, small b. Now here, when all four are dominant, see all either this A, this A, this B, or same have all have same contribution, and recessive is not going to contribute anything. So when four dominant allele is there, then they are going to produce a dark red. Which phenotype will be obtained? Dark red. Now when out of four, if any one get recessive like this, here B is getting recessive, here you can see in case of here A is getting recessive. So this both will have a same phenotype. Why? Because three are dominant in this. So when there is a three dominant allele, the phenotype will be somewhat lighter red, and after this two dominant allele are there. When there is a two dominant allele, then intermediate red is obtained, and when one dominant allele, it is either A or B, It doesn't matter that genotype with the one dominant allele is going to produce very light red, and when zero dominant allele, it is not going to give a slight radiation as well, so phenotype is going to be white. So if you can see here, four dominant are there, so here you will write a dark red. Okay, here dark red I am writing like this. So here three recessive are there, so it is a light red. Here one recessive is there. Here three dominant and one recessive, so here it is also light red. But here two are getting recessive. Then when on two are remained as a dominant, then it is an intermediate red. Okay. Then here also three are dominant, so here they are light red. Here two are dominant, so they are intermediate red. Here two are dominant, so they are getting intermediate red. Here one is dominant only, so then it is a very light red. It is a very light red. Here also three are dominant, so here it is light red. Two are dominant, so it is intermediate red. Two are dominant, here it is also intermediate red. And here one is dominant, so here very light red. Here two are dominant, so it is intermediate red. Here one is dominant, so it is going to be a very light red. Here also one is dominant, so very light red. Here, if you see, then here all are recessive, so it is a particularly we can say a white. So if you see how many dark red are there, so dark red is one, white is one, but light red are one, two, three, four. Okay, so if we take a phenotypic ratio, for dark red is 2, light red is 2, intermediate red is 2, very light red is 2, white, then dark red are 1, light red are 4, you can count in this one, intermediate red are 1, 2, 3, 4, 5 and 6, very light red are 4 and white is also 1. So the phenotypic ratio of this polygenic inheritance, and when two genes are showing a polygenic or quantitative inheritance, then it is going to give a phenotypic ratio that is 1 is to 4 is to 6 is to 4 is to 1. And if we draw a particular curve for this, then you can see that this can produce this type of particular phenotype curve. Here it is representing 1 by 16, then 4 by 16, 6 by 16, 4 by 16 and 1 by 16. If you see this phenotypic ratio and you draw a curve, you will see this bell-shaped curve is obtained. So this is for the two gene. If they show polygenic inheritance, then this phenotypic ratio will be obtained in F2 generation. But when there are a more than two gene, then we have to discuss that also. So for more than two gene, we are going to take some of the example and we will discuss one of them for this. So other example of character which show polygenic inheritance. And if we discuss other example, the other examples are human skin color. This human skin color is controlled by three genes. Then second, human height. It is controlled by 5 genes. Third, human intelligence. That is also controlled by a polygenic inheritance. And this human intelligence show a 25 genes. Then if we take another example, then milk production in cattle. That is also controlled by a polygenic inheritance. Then if we see another example, then meat production in the animal is also controlled by polygenic inheritance or multiple gene. And sixth, shape, size, and number of fruit and seeds of many plants are controlled by many genes, means that is also example of polygenic inheritance. But in that, we are going to discuss human skin color in detail, because sometimes you will get a question from this part. So we have to discuss a human skin color inheritance according by the multiple gene, and it is controlled by 3 pairs of genes. Okay, so let's understand the human skin color, polygenic inheritance of human skin color character. It is regulated or controlled by 3 pairs of gene, but it is studied by Davenport, and it is controlled or regulated by 3 pairs of gene. So total 6 genes are involved here. Okay, 3 pairs of gene, like what? Gene A, so it can be either in capital A form, or capital A form, or small a form. So another B gene, that can be in a capital B, capital B small b, or small b. And another one is the, so that third gene can be capital C, or capital C, or small c. Means 3 genes are there, and each gene have their own allele. So total 6 genes are going to be there. Okay. So what are that gene and how they contribute? So here you will see, when there is a, all are in a dominant form, they are going to give a very dark phenotype, because all gene are contributing in the melanin production, all the 6. But when all get recessive, then they are not going to produce a melanin, so they are going to be very light. Okay. So if we want to make a cross of this pure and understand how they show the polygenic inheritance and what will be the phenotypic ratio, then let's understand by this particular cross. Here the complete cross is made, that we need to understand. See here, you can see that this is the parent generation. In parent generation, white plant is taken, that is a completely white, okay, very light, that is a small, sorry, very light, we can say color of a parent. And very dark color when there is a maximum melanin production, that is capital A, capital A, capital B, capital C, capital C, capital C. The gametes will be going to be small a, small b, small c. Here's capital A, capital B, capital C. When they fuse, they form a capital A small a, capital B small b, and capital C small c. Now it has only 3 in a dominant, So exactly half production of mineral will occur. So that is the exact intermediate between the a very dark and very light. But now this when undergoes selfing, when we carry out selfing of this, the F2 generation will be obtained, and this is the result of this F2 generation. In this gamete will be like this: capital A, capital B, capital C. 8 gamete will be produced. See, 3 heterozygous condition are there. So 2 power n, 8 gametes will be produced, and 8 gametes will be produced like this. And I have already taught you in the how the gamete formation occur for this 3 heterozygous condition. Same way, 8 gamete will be of here for the another hybrid. And when we make a selfing of this, you will see the 64 combinations. When we make a cross of this all 8, you will see this particular genotype. Okay, so you know how to write a genotype that you are able. You can make, just take this particular genotype and make a cross with this one. So when capital A, capital B, capital C make a cross with the capital A, capital B, capital C, it is going to give rise to the capital A, capital A, capital B, capital B, capital C, capital C. So we multiply all this with all 8, then multiplication of this all with all this, then cross of this one, this gamete with fusion of this gamete with all these gamete. Same way, these all gamete are going to fuse one by one with this gametes. When all are in a dominant form, that is going to produce a very dark. Okay, but when one get recessive, either it is A, either it is B, either it is C, it is going to be dark because some darkness will be going to be reduced. So see here, A is recessive, then it is also dark. Here B is recessive, then it is also dark. Here C is recessive, then it is also dark. Means ultimately the requirement is what? Here 6 are dominant alleles. If the 5 dominant gene are there, then it is going to be dark. Then when out of 5, 4 getting particularly a dominant and 2 get recessive, then it is fairly dark. You can see here A and B are recessive. Okay, here small a and small b is recessive, so it is going to be fairly dark. Here it is a wrong mistake. Here should be capital A, capital A. Here they have written capital A, small a. So capital A, capital A, capital B, small b, small c, small c. So here B and C are recessive. 4 are dominant, so that is fairly dark. Here also we can say 2 are recessive and 4 are dominant, so fairly dark. But when out of 4, 3 remain as a dominant and 3 get recessive, then it is intermediate. Out of 6, after 3, if 2 become remain dominant, then it is fairly light. Either it is AB, AC, AB, whatever, anything. If 2 are dominant and 4 are recessive, then it is going to be fairly light. Then one get recessive, then it is going, one get dominant, one remain dominant, and other all get recessive, then it is a light. And when all get recessive, it is very light. So what you will see here, as the number of dominant alleles are getting reduced, the darkness is going to reduce because melanin production is going to be reduced. So if we consider a very dark, when all the 6 genes are or 6 alleles are dominant, then it is going to be very dark. When 5 are dominant, then it is going to be dark. When and very dark are 1. When 5 are dominant, 6 are going to be dark. When fairly dark, then 4 are dominant and fairly dark are 15 genotype are obtained. 15 phenotype are obtained. In intermediate, if we see, then 20 strings are obtained. Fairly light, here 3 are dominant, here 2 are dominant, then fairly light are 15. And here light means 1 is dominant, then it is 6. When all are recessive, no dominant, 0 dominant, then it is going to be white. So ultimate phenotypic ratio is 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. That is a phenotypic ratio of human skin color inheritance through the polygenic inheritance. And if it occurs like this, if we make a curve of this, then for curve also you can draw like this histogram 1 by 64, then 6 by 64, 15 by 64, and 20 by 64. So when you see, if you make a histogram, then also you can make a histogram like this. Okay, so this is 1 by 64 is nothing but what? Very dark, then it is dark, it is fairly dark, it is intermediate, it is fairly light, it is light and very light. If we draw a same, then you can see like this bell-shaped curve. This is a histogram, and same way bell-shaped curve can be obtained like this. So this gives a histogram or bell-shaped curve are obtained in a polygenic inheritance. This is also named as normal distribution curve. Okay, so this is the thing which is obtaining this. So you can see here when three pairs of genes are controlling the character, the phenotype are seven. Okay, seven phenotype are obtained like the very dark, dark, fairly dark, intermediate, fairly light. Total seven phenotype are obtained. So there is one formula, then total how many phenotype are going to be present in this, that we can conclude by using one formula. So write that formula here also. If the character is controlled by polygenic inheritance, we can use this formula and identify that how many total possible phenotypes are there. So for this, the formula for the total possible phenotype is 2n plus 1, where n is equal to number of genes. So this is the total possible phenotype. And to identify total possible genotype, the three pairs of gene are here. The formula is 3 power n, where here n is equal to number of genes. Only genes, not in pair. Okay, only genes we are going to consider. So three genes are there. 3 into 3 into 3. So it is going to produce a what? A particular 27 genotype are possible. Okay. So this formula are obtained. So if six genes are responsible for in a dominant form for the maximum expression, and then all six are in the system, then they are for the minimum expression. Okay. Then if we want to identify the contribution of each dominant allele, then we can use one more formula. Contribution of each dominant allele, then it is identified by using a formula, that is maximum expression minus minimum expression upon number of dominant allele. So how to use this all formula, and means mainly the third one, to understand that we have to take some example, or we have to make some questions regarding the polygenic inheritance, then only we can find out how to use that third formula. So for that, let's discuss some of the questions. And in that, we are going to discuss the first question that says that a plant is homozygous for AB gene. have 20 feet height, while the minimum height of plant is 6 feet. Then find out the following. Number one, contribution of each dominant allele. And second, calculate the height of plant with following genotype. The genotype are capital A, small a, capital B, capital B, capital A, capital B, small a, capital B, small b. Okay, so this thing we have to identify. So let's solve these two questions. If we want to identify contribution of dominant allele, the formula is going to be what? Maximum expression minus minimum expression upon number of dominant allele. So if we see the homozygous for AB, that is capital A, capital A, capital B, capital B, the given is that is have a height 20 feet. And the smallest, small b have the height that is 6 feet. So if we calculate this thing, the maximum expression is 20 minus minimum expression is 6 and upon total dominant gene if we say that are 4. So if we calculate this 20 minus 6 is going to be 14 and 14 divided by 4. Okay, 20 minus 6 is going to be 14. 14 divided by 4. So if we divide 14 divided by 4, then it is a 3.5 feet. Why? Because the recessive has no contribution that we have studied. So 35.5 feet is contributed by each dominant allele. Either it is capital A or capital B. Okay. So if we know the contribution of dominant allele, then that dominant allele is going to contribute above the minimum height. Yes or no? Because minimum height is going to be 6 feet. If whatever the dominant allele increasing in that case, that will contribute to this particular increase in height. So if capital A, capital B, small b, then the minimum height is 6. But how many dominant are there? 3 are dominant. So we multiply 3 by the 3.5. So if we multiply 3.5 with the 3, then it is going to be 6 plus 3 3s are 9, 10.5. So it is going to be 16.5 feet height with the genotype. When there are two dominant, then it is going to be 6 5, 6 plus 5 into 2 into 3.5. So it is going to be 6 plus 7. So it is 13 feet. Then when the genotype is with the one dominant, then 6 plus 3.5. So it is going to be 9.5 feet. Then there is a one dominant allele. So like this, you are going to calculate this, the contribution of each dominant allele and the height of the plant with the particular genotype. Okay, so do that, to do that or understand this one more detail, we are going to discuss one more question. And another question is, so let us discuss another question. For tri-hybrid plant, tri-hybrid plant, that is capital A small a, capital B small b, capital C, capital C small c, produces fruits of 70 grams weight, while minimum weight of fruit is 10 grams. So then find out the following. Number one, contribution of dominant allele. Second thing that we need to identify, that maximum weight of fruit. And third, we need to calculate weight of fruit of following plants, and that is capital A small a, capital B, capital B, capital C small c. And second, capital A small b, capital C small c. Okay, we have to identify the fruit weight of this. So if you want to identify a contribution of dominant allele, then we need to identify a maximum expression minus minimum expression. But here, this is given. So here, the maximum expression, if we identify for the capital A small a, capital B small b, capital C small c, for that particular trihybrid genotype. So for the trihybrid genotype, when 3 are dominant, the maximum expression is 70, minimum expression is 10, and total dominant allele are how many? 3. By taking this genotype, we are calculating. So 3 are dominant gene, 1, 2, 3. So it is going to be 60 by 3. So 20 gram is the contribution of each dominant allele. So if you want to identify maximum weight of food, then the genotype is going to be capital A, capital B, capital B, capital B, capital C, capital C. So here how many? 6 are dominant. So if we multiply 6 with 20 gram plus minimum weight is 10, so this is going to be 130 gram. That is maximum weight of the fruit, because minimum weight is 10. Above which, if then there is a 6 dominant, the dominant contribution of dominant is 20 gram, so it is going to be 130. And if you identify a the particular weight of this fruit, then the minimum weight is 10 plus how many? 1, 2, 3, 4. 4 are dominant, and each dominant have 20 contribution. So 24s are 18 plus 9, 10. So it is going to be 90 gram. Same way here, the 10 is minimum, and here 1 and 2 are dominant. Each has a 20 contribution. So 20 is to 40, 40 plus 10 is a 50 gram. Okay. So like this, you can apply the knowledge of polygenic inheritance on this type of question. Okay, dear students. So I hope you understood it in a very well that what is polygenic inheritance, what is maximum by expression, why it is called as quantitative inheritance, why it is known as particularly we can say quantitative inheritance. Okay, so this all multiple gene interaction, that all thing we have discussed in this lecture. Okay, so dear students, I hope you understood that. Thank you.